Thanks, Laura. Yeah, all of the sessions today were really fantastic, and I was asked to sort of summarize the second session a little bit. Looked better on my laptop. So for the case studies in precision drug development, I wanted to highlight four things. One is the creative trial design. It was really exemplified in the two cancer examples, but I think we should really extrapolate that to everything we do. I was very impressed with the recruitment strategies that we saw, where diagnostic tests could be performed anywhere for the trial that Zach described, and the sponsor was able to take trials in. And, I really like the just-in-time site establishment. For rare diseases, this is kind of key because you just don't know where you're going to find people. Those are really nice advances that I think everyone should sort of take a close look at. We talk about collaboration a lot, and the lung map study was a great example of, multiple academic centers, multiple companies. And I think just building on something Laura mentioned, there may be an opportunity here for a new business model. Having done a lot of collaborations from a company, I can tell you that when the drug sponsors take on too many responsibilities, things get bogged down. When you have a situation where the drug company is providing money and there's a third party, whether it's an academic group, a CRO, things are better. So one possible new business opportunity here, and something we should all think about, is how do we get an entity to do something like collect all the genetic data, centralized phenotypic data, so that the sponsors can access it in a way that they're comfortable with. Maybe the patients have a little bit more ownership, but it doesn't rely on company X, Y, or Z doing too many things, because company X, Y, or Z may change priorities tomorrow. The other thing we saw is there's complexity even in Mendelian diseases. It all seemed so simple when we started working on SMA. My colleague made a great talk earlier. But there's multiple types of SMA. There's more tests that need to be done after that initial test. So there's a lot of patient heterogeneity, even in single gene disorders, that we need to deal with. And then, there's locus heterogeneity. And I think PJ highlighted one approach to locus heterogeneity in rare diseases, but exemplified by the mitochondrial disorders, you have a lot of different loci that cause similar diseases. It's probably not cost effective to make individualized therapies for each one of those mutations. And we've all talked a lot about the pathway approaches, but it's very hard to identify the common intervention points. So there's complexity everywhere, even in Mendelian diseases. And again, I think really through more data sharing is probably the best way to get the most out of that. And then we saw advantages of pre-symptomatic treatment in SMA, where it's pretty dramatic. And how do we go beyond pediatric diseases? People need to be willing, if they know they're at risk, to get into a trial. We need to figure out how to make that happen. So, from those case studies, what we saw, and as organizers, we attempted to group together cancer and rare diseases, because on the surface they seem easier. We realize they're harder, but they do provide some examples for more common disease as well. And with that, the next speaker is Russ.